Muscle Physiology, Chapter 1. This chapter provides an overview of how muscles work to create forces across joints and to cause movement. Understanding the physiology of muscles is basic to developing and implementing training programs. At the simplest level, the strength and conditioning professional is concerned with the function of muscle. Many systems, including cardiovascular, nervous, and endocrine systems, affect the function of muscle. But muscles is the organ that creates movement. In other words, the other systems affect performance by changing how the muscle functions. Skeletal muscle, macrostructure, and microstructure. Each skeletal muscle is an organ that contains muscle tissue, connective tissue, nerves, and blood vessels. Fibrous connective tissue, or epimysium, covers the body more than 430 skeletal muscles. The epimysium is continuous with the tendons at the ends of the muscles. The tendon is attached to the bone periosteum, a specialized connective tissue covering all bones. Any contraction of the muscle pulls on the tendon and, in turn, the bone. Limb muscles have two attachments to bone: proximal, closer to the trunk, and distal, further from the trunk. The two attachments of the trunk muscles are termed superior, closer to the head, and inferior, closer to the feet. Traditionally, the origin of a muscle is defined as its proximal, towards the center of the body, attachment, and the insertion is defined by the distal, away from the center of body, attachment. Muscle cells, often called muscle fibers, are long, sometimes running the entire length of a muscle. Cylindrical cells, 50 to 100 micrometers in diameter, about the diameter of a human hair. These fibers have many nuclei situated on the periphery of the cells and have striated appearance under low magnification. Under the epimysium, the muscle fibers are grouped into bundles, fasciculi, that may contain up to 150 fibers, with the bundles surrounded by connective tissue called perimysium. Each muscle fiber is surrounded by connective tissue called endomysium, which is encircled by and is continuous with the fibrous membrane, or sarcolemma. All the connective tissue, epimysium, perimysium, endomysium, is continuous with the tendon, so tension developed in the one muscle cell is transmitted to the tendon. The junction between a motor neuron, nerve cell, and muscle fibers is that it innervates is called the motor end plate, or more often, the neuromuscular junction. Each muscle cell has only one neuromuscular junction, although a single motor neuron innervates many muscle fibers, sometimes as many as several hundred. A motor neuron and the muscle fibers it innervates are called a motor unit. All the muscle fibers of a motor unit contract together. When they are stimulated by the motor neuron, the interior structure of a muscle fiber is depicted in Figure 1.3. The sarcoplasm, the cytoplasm of a muscle fiber, contains contractile components, which consist of protein filaments, other proteins, stored glycogen, and fat particles, enzymes, and specialized organelles, such as mitochondria and sarcoplasmic reticulum. Hundreds of thousands of myofibrils, each 1 micrometer in diameter, 1/100th the diameter of a hair, dominate the sarcoplasm. Myofibrils contain the apparatus that contracts the muscle cell, which consists primarily of two types of myofilament, myosin and actin. The myosin filaments, thick filaments, 16 nanometers in diameter, about 100,000th the diameter of a hair, contain up to 200 myosin molecules. Globular heads, called cross bridges, protrude away from the myosin fibers, myosin filaments, at regular intervals. The actin filaments, thin filaments, 6 nanometers in diameter, consist of two strands arranged in a double helix. Myosin and actin filaments are organized longitudinally in the smallest contractile unit of skeletal muscle, the sarcomere. Sarcomeres average 2.2 micrometers in length in a relaxed fiber, approximately 4,500 per centimeter of muscle length. and are repeated the entire length of the muscle fiber. Figure 1.4 shows the structure orientation of the myosin and actin in the sarcomere. Adjacent myosin filaments anchor to each other at the bridge, at the M bridge, at the center of the sarcomere, the center of the H zone. Actin filaments are aligned at both ends of the sarcomere and are anchored at the Z line. Z lines are repeated throughout the entire myofibril. 6 actin filaments surround each myosin filament, and each actin filament is surrounded by 3 myosin filaments. It is the arrangement of the myosin and actin filaments and the Z lines of the sarcomeres that gives skeletal muscle its alternating dark and light patterns, its striated appearance under magnification. The dark A band corresponds with the alignment of myosin filaments, whereas the light I band corresponds with areas in 2 adjacent sarcomeres that contain only 1 actin filament. The Z line is in the middle of the I band and appears as a thin, dark line running longitudinally through the I band. The H zone is the area in the center of the sarcomere where only myosin filaments are present. During the muscle contraction, the H zone decreases as the actin slides over the myosin towards the center of the sarcomere. The I band also decreases as the Z lines are pulled toward the center of the sarcomere. Parallel to and surrounding each myofibril is an intricate system of tubules called sarcoplasmic reticulum that terminates as vesicles in the vicinity of Z lines, figure 1.3. Calcium ions are stored in these vesicles. The regulation of calcium controls muscular contraction. T tubules, short for transverse tubules, run perpendicular to the sarcoplasmic reticulum and terminate in the vicinity of the Z line between the 2 vesicles. This pattern of T tubule, spaced between the perpendicular to 2 sarcoplasmic reticulum vesicles, is called a triad. Because the T tubules run between outlying myofibrils and are continuous with the sarcolemma at the surface of the cell, discharge of an action potential, an electrical nerve impulse, arrives nearly simultaneously from the surface to all depths of the muscle fiber. Calcium is thus released throughout the muscle, producing a coordinated contraction. In its simplest form, the sliding filament theory states that the actin filaments at each end of the sarcomere slide inward on myosin filaments, pulling the Z lines towards the center of the sarcomere and thus shortening the muscle fiber. As actin filaments slide over myosin filaments, both H zone and I band shrink. The flexion of myosin cross-bridges pull on the actin filaments and is responsible for the movement of the actin filament. Because only a very small displacement of actin filaments occurs with each flexion of the myosin cross-bridge, very rapid repeated flexions must occur in many cross-bridges throughout the entire muscle for measurable movement to occur. Under normal resting conditions, little calcium is present in the myofibril. Most is stored in the sarcoplasmic reticulum, so very few of the myosin cross-bridges are bound to actin. No tension is developed in the muscle, so the muscle is said to be at rest. Before myosin cross-bridges can flex, they must first attach to the actin filament. When the sarcoplasmic reticulum is stimulated to release calcium ions, the calcium binds with troponin, a protein situated at the regular intervals along the actin filament that has high affinity for calcium ions. This causes a shift to occur in another protein molecule, tropomyosin, which runs along the length of the actin filament in the groove of the double helix. The myosin cross-bridge heads now attach much more rapidly to the actin filament, allowing cross-bridge flexion to occur. The energy for cross-bridge flexion comes from hydrolysis, the breakdown of adenosine triphosphate, ATP, to adenosine diphosphate, ADP, and then phosphate, a reaction catalyzed by the enzyme myosin ATPase. Another molecule of ATP must replace the ADP on the myosin cross-bridge of the head to detach from the active site and recock. This allows the contraction process to be continued if calcium is available to bind to the troponin, or relaxation to occur if calcium is not available. It may be noted that calcium plays a role in regulating a large number of events in skeletal muscle besides contraction. These include glycolytic, oxidative, and energy metabolism, as well as protein synthesis and degradation. Measurable muscle shortening transpires only when the sequence of events binding the calcium to the troponin, coupling of the myosin cross-bridges with actin, cross-bridge flexion, dissociation of actin to myosin, and recocking of myosin cross-bridge head is repeated over and over throughout the muscle fiber. This occurs as long as calcium is available in the myofibril, ATP is available to assist in uncoupling the myosin from actin, and sufficient active myosin ATPase is available for catalyzing the breakdown of ATP. Relaxation occurs when the stimulation of motor nerve stops. Calcium is pumped back into the sarcoplasmic reticulum, which prevents the link between actin and myosin filaments. Relaxation is brought about by the return of actin and myosin filaments to their unbound state.